Okay, this video is a review or an overview of how nephrons work. So this first image is from probably your textbook or else another textbook, and it has a simplified version of a nephron. I like it because it has things separated into simple stages. First, we have blood coming in through the afferent arteriole, heading into the glomerulus, leaving through the efferent arteriole, and heading to the capillaries that are wrapped all around this. But this diagram is nice and simple. It doesn't make it messy like that. So the filtrate comes through holes in the glomerulus. It's like plasma, but it's not in your blood anymore, so we don't call it plasma, we call it filtrate. All of the big things like red blood cells, white blood cells, platelets, they're going to continue on this ride and keep in your circulatory system. They're not going to enter the nephron at all. That includes everything up to big proteins. Smaller stuff goes through here, and we got a couple of stages here. Waste being added in, but first useful things being reabsorbed. Reabsorbed mean going back into your body. It was in your body, now it's going back. Secretion means going into this tube. This tube is headed to the toilet. These are things we want to get rid of. These are waste products. Both of these are happening throughout the trip through the nephron. At the end of the nephron is our last chance to balance water, lots of water being reabsorbed in the collecting ducts. So let's take a look in a little bit more detail over here. So we have our nephron, and our nephron starts, just like we said, with blood coming in through the afferent arteriole and heading into the glomerulus, where it's filtered by size. Filtering by size means that we don't lose any big important things like red blood cells or white blood cells. As we go through, our blood goes through here, it's eventually going to leave out the efferent arteriole, and then it's going to wrap all around the tubes, but I didn't draw that because I had enough of a struggle drawing this. The next section, or the first section of the actual nephron, is the cup that collects all of that filtrate. That cup is called the Bowman's capsule. Bowman's capsule is just based on somebody's name. It's just clear to collect the filtrate. Nothing special happens there, no absorption or secretion, just collection. Then our filtrate heads into the first section of our nephron, called the proximal tubule or the proximal convoluted tubule. The proximal convoluted tubule has a lot of jobs, a lot of things that we don't mention in Bio 20, but we need to remember the most important thing is that it reabsorbs, so it keeps things like sugar and amino acids. This, if you remember nothing else about the proximal tubule, this is what I hope you remember. It does a lot of other things, like it gets rid of some urea and uric acid. Some drugs get secreted at that point, make it in your urine. Things like small proteins that made it past the glomerular filter, things like vitamins, lactate, which we know is a byproduct of fermentation, some water, some ions, they all have their chance. The proximal tubule, we said, was a lot like our small intestine, and our small intestine is long, twisty, has lots of microvilli, and the microvilli, the twistiness, and the mitochondria to power all of this results in opportunity for a lot of active transport. The next section, as we come past the proximal tubule, we're going to be down, down, down into the descending loop of Henle. The descending loop of Henle makes a stop, makes a transition between the cortex above and dips down into the tissue of the medulla. And we know that there's a saltiness gradient, that as we go down towards the medulla, things get saltier. That helps to draw out lots of water. The descending loop of Henle is permeable to water. That means water can go through the holes in the side, but not salts. That means that water is going to be drawn out because it's salty down here. And the salts have to stay inside. The concentration of the salt inside here is getting saltier and saltier and saltier outside and inside of this tubule. But as it heads back up into the ascending loop of Henle, so we're up here in the ascending loop of Henle part, this situation changes. It becomes impermeable to water. These walls are thicker, less likely for water to get through, but salt gets pumped out actively. So sodium ions, chloride ions, potassium ions, if you remember one, remember sodium, gets pumped out actively, and those salts getting pumped out actively, things like sodium, will increase the saltiness, the solute concentration, the pull that this area has towards water more and more and more. That makes this step of taking water out easier and easier, which is great. We need so much water, and osmosis is the cheapest way to get it. We don't want to waste it. Then we head through the next twisty section, or the last twisty section here, called the distal convoluted tubule. Distal means far away, proximal means close to the point of connection. In the distal tubule, we're maintaining the homeostasis of ions. Lots of ions, more than what I've listed here, but again, this is what you need to know. Potassium ions, sodium ions, chloride ions, hydrogen ions, and bicarbonate ions. In particular, we need to remember two of these sets. One is the sodium and chloride set. Sodium levels are important for proper nerve function, and aldosterone is the hormone that we use when we have low sodium in our blood. If we need to reabsorb more sodium, if we need to keep more sodium, we make aldosterone. This happens in the hypothalamus in your brain. There are osmoreceptors there that measure the saltiness of your blood. If you need more sodium, you secrete more aldosterone. More aldosterone means that more sodium ions are going to make it out of the pathway that's leading to the toilet, so we get to keep them. The other set that we need to remember is about hydrogen and bicarbonate ions, which work together to do balancing of pH, keeping your blood pH consistent and neutral. If your blood is too basic, that means you need more acid in your blood to neutralize. So we would reabsorb H+, which is an acid, and secrete bicarbonate ions. These always go, these two ions, hydrogen and bicarbonate, always go in opposite directions. If you're secreting one, you're absorbing the other. If you're absorbing one, you're secreting the other. The normal case, though, is that your blood is too acidic because you've got CO2, and that's the thing that's most likely to build up in your blood, in which case you would secrete hydrogen ions and reabsorb bicarbonate ions. This helps to bring your pH up towards a more basic level or a more neutral level, and so does this. The last stop is the collecting duct. The collecting duct is shared between many nephrons. The collecting duct has little holes in it called aquaporins. They can open or close. The hormone ADH will make them open. The hormone ADH is, again, regulated by the hypothalamus in your brain. The osmoreceptors there say, hey, there's not enough water, we need to conserve water. Let's secrete this hormone ADH. That opens up these pores, and water can come out by osmosis. Water comes out by osmosis because it's salty. It's not just salty right there. I mean, it's salty everywhere down in the medulla. There's lots of nephrons in this whole area. This is a quick look at the things you need to remember for your nephron. I'm just going to come over to this image as well. And this image, just for context, if we're looking at a kidney, we have the cortex tissue and the medulla tissue. The nephron dips between both. The loop of Henle dips down into the medulla and back up. And although our diagrams don't make it look that way, they dip a long way down and back up. They take a long trip down into that salty area. It's not like a millisecond pathway. They're down there in a while, which just means they have a really good opportunity for this passive reabsorption of water. Reabsorption of water, maintenance of water levels is super critical for your body systems. I hope this helps.